This episode is supported by two companies directly invested in advancing GPCR research, Wnt DiscoverX and GeneTex. Wnt DiscoverX brings decades of validated GPCR assay expertise to teams building real drug discovery programs. It's trusted across discovery, development, and regulatory review. And GeneTex believes antibody trust is earned, not claimed. GeneTex believes antibody trust is earned, not claimed, proving it through rigorous validation and an open invitation to the community to test it, the community being you. You get to challenge and give feedback. Request your free anti-GPCR antibody sample today. Links to both are in the show notes. And now, let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR. It is my great pleasure of having with me Professor Gunnar. See, I did mix it up, but we were just talking about how to pronounce their name. So let me do that again. Gunnar Schulte. I am very happy to welcome you, and you're from Karolinska Institute in Sweden. Hi.
Hi. Thank you very much. Thanks for the opportunity to be with you.
I'm excited to have you with us today, especially that you work on a very specific class of GPCRs about which we haven't recorded yet a podcast episode, nor did we have a guest that worked on this class of receptors. But before we dive into that, can you please introduce yourself to us and tell us a little bit more about your career?
Yes, I will do that. So my name is Gunnar Schulte. I'm a professor now at Karolinska Institute, and I have an undergraduate education in biochemistry from the Free University in Berlin. So I'm from Germany. I'm born and raised in Germany and started studying biochemistry in something like, must have been 92, with a genuine interest in natural sciences. And the interesting part of the biochemistry studies in Berlin was that it was a very small group of students every year. So I think we were 24, and we were encouraged to look abroad for practical works, for laboratory rotations outside the university at institutes. And my plan was actually to go to Brazil, to a former colleague of my father, to the University of Campinas. And the day I was about to book my ticket, the family I was supposed to live with, they called me and said they had been robbed with machine guns the third time in a row, and if I might reconsider. And I honestly reconsidered, and I sent a bunch of applications for a rotation to Sweden, to Stockholm, to the Karolinska Institute. And there was one of, I think, 6 or 7 applications that was positive, and I got invited for a 3 months stay. No salary, just labbing at Karolinska. And that was the start of what is now 25 years in Sweden.
Wow, that's so interesting. So first of all, let's just take a step back to Berlin and back to Germany. I really want to hear more about your choice of trying to go to Brazil. But also you mentioned that you were supposed to go to Brazil for an internship to work in your father's lab. So two questions out of that. One is, when did your interest in science when did you realize you were interested in science? And is anyone else in your family a scientist?
Well, my father is a chemist, and I think that was initiating the interest. So already as a young child, I was interested in minerals, and I was categorizing minerals and their hardness and color and whatever semi-scientific stuff you could note as a 6-year-old. And I was always interested in fish and fish biology and limnology and marine biology. So my idea was to study limnology or marine biology, which for some reason didn't really turn out. I tried to, but I got a spot in Berlin for biochemistry instead, which I'm, in hindsight, really, really thankful for.
Interesting.
But
and how come?
It was not my father's lab in Brazil, but it was a friend of his,
Friends, yes.
who he studied with. Exactly.
So, but then since you ended up in the biochemistry field, why not chemistry?
Well, it was chemistry was the start of it was actually a very, very chemically oriented. The bio and the chemistry came very late, and I since my love to biology was always apparent already as a child, I really didn't want to go the organic chemistry school that my father went, but rather turn to the biochemistry part.
That is interesting. And during your PhD in Berlin, did you have any practical, any lab experience that you can gain? Or how is, I'm asking this is because depending on the country you're in, the PhD program can be very different. And I was wondering, how was your PhD program at the time?
Before we continue, I want to ask you something. A lot of you listen regularly to the podcast, but haven't subscribed yet. If you enjoy these conversations, hit subscribe now and leave us a review. It helps other GPCR scientists find the podcast. And speaking of community, a couple of things worth mentioning before we continue. The Dr. GPCR University is where GPCR training meets a real scientific mission, expert-led, live and on demand, and built to move the field forward. All the links are in the show notes. And now let's get back to this episode.
Well, the in Berlin was actually the undergraduate studies, which you would call now a master's, I would guess. But also there it was very practically oriented. So we had protein chemistry and really practical labs, and I think that is one of the good things in the education still today, that is very, very practically organized. And you have even rotations in Berlin. I mean, in Berlin you have fantastic institutes and laboratories where rotations are possible already at very young career age. So I went actually then to Sweden before I finished my master's studies, so my diploma thesis.
Okay. And then where did the idea of potentially going to Brazil come from? That's a long way from Berlin.
It's a long way. Yeah, well, it's somehow botanizing in attractive geographic localizations with the connection where you probably could, I could stay for free, even though it sounded very dangerous. Yeah. And it was an interesting, this was organic chemistry, but it was an interesting practical work for the 3 months.
Interesting. And then when, since you reconsidered considering the safety issues in Brazil, and you ended up in Stockholm, what did you work on? You mentioned that you had submitted multiple applications and you had to pick one that you liked. What did you pick, and what motivated you to pick that?
Yeah, so in at that stage of my career, I was interested either in virology or in signal transduction research. And I sent out applications for both, and it turned out to be a neuroanatomy, neuroscience, spinal cord research project, which was basically immunostaining of rat spinal cord sections for the time being, which was kind of cool in a way that was the first time for me as biochemistry to dive into medically relevant bioscience.
That's so interesting. And then after the 3 months, what came next? Did you decide you wanted to pursue a PhD, or?
In Berlin, I had the opportunity to work with a small company that is, I think it's still around. It's called Timolbiol or something like that. They had the license to synthesize peptide nucleic acid in Europe. Mm-hmm. And I think I was one of the first ones who actually did that on their synthesizer. I tried to earn some money aside. And I took that interest in that novelty to Stockholm and tried to set up an in situ hybridization in spinal cord tissue with peptide nucleic acid. And I think I had a postdoc at the SFN meeting, but it never really got further than that. But that meant that I could stay another 3 months. And after that, I had actually to go back to Berlin to finalize my exams to really finish up with the studies in Berlin.
Great. And then where do GPCRs enter the picture at this point in your career?
Yeah, they enter exactly at this point. So my supervisor introduced me to my PhD advisor, Wnt-frizzled. And the reason was that I heard a talk about extracellular ATP being a signal molecule. Mm-hmm. And since I only knew ATP as a part of DNA or also energy currency, I was interested. And there was this professor working at the Karolinska Institute. We're not at ATP receptors, but adenosine receptors. Yeah. So, and I got somehow interviewed with him and started my, at that time, diploma thesis or master thesis, but continued then also my PhD project with him.
Wow, interesting. So you're kind of doing things in parallel once you learn about the adenosine receptors. And how did you choose the projects that you were working on? Was it something that the lab was already working on, or did you have any specific ideas on what was interesting to you?
Yeah, that was the lab was very much caffeine heavy. So they looked at the effects of caffeine and in brain and on immediate gene responses. And some new aspects that a student before me had already started was really signaling using phospho-specific antibodies and mapping signaling pathways. And I really dived into that and worked on adenosine receptor signaling to mitogen activated protein kinases, really biochemically mapping signaling pathways.
So that's so interesting. I was just listening to a podcast from this neuroscientist from Stanford, and he was talking about, so he's basically, the whole idea about his podcast is translating scientific discoveries into everyday tips and tricks on how to be better and more productive in your life. And he was just talking about the adenosine receptors, and was mentioning that it's a good idea if you want to increase the effect of your coffee, you should delay taking that coffee by 2 hours in the morning, because the caffeine actually displaces the natural adenosine from the receptor, the adenosine ligand from the receptor. And if you wait those 2 hours, then the effect of the coffee is greater and it lasts longer. Which I thought was a very interesting thing. And then what took you from adenosine receptors to the GPCR family that you're working on currently?
Yeah, so I defended my thesis on adenosine receptors in '96. Is that true? No, I started in '96 and I defended in 2002. So after that, I was up for a postdoc, and it actually did not take me very far, only a few buildings from where I worked before. So I was at the Department of Physiology and Pharmacology, and then I went to the Department of Medical Biochemistry and Biophysics. And there was a Professor Ernest Arenas, who had been already in my half-time advisory board for the thesis before. And he was working on this signaling pathway where Wnt are the ligands, Wnt, and frizzles are the receptors. And somehow we got together, and I got a postdoc position there to investigate Wnt and frizzle signaling in the midbrain, with relevance to development of midbrain dopaminergic neurons.
And then from your postdoc, and it's interesting. So let's just pause a little bit, because most of the time they tell you when you should be moving away from the institute or from the place where you do your PhD to do a postdoc. How come you just switched just a couple of buildings over?
Well, my wife at that time, now my ex-wife, she was around. We had actually a positive pregnancy test on the day of my thesis defense. So moving was not really on our plans for that time.
Got it. Was it before, and you don't have to answer, but was it before or after you defended your thesis?
Well, it was actually the evening after the thesis defense, or the morning after the thesis defense party.
Okay,
so pregnancy test.
at least you were able to focus on the thesis defense on that day, because those days tend to be pretty stressful. Oh, that's great.
That's a good day. Yeah.
That's great. And then so you started working on Wnt ligands and their receptors of the F family. How did you decide to continue working on the frizzled receptors long term?
Yeah, I was the honest question is I was not satisfied about what's known about those. I mean, I had somehow figured out what questions to ask and what experiments to do, but the toolbox was so limited that I somehow saw an opportunity there to continue after that postdoc. Well, I have to say the path was not very straightforward at the time. I took a few detours before I landed really in solidly looking and working on frizzles.
So tell us a little bit more because the reason I'm asking this is because most of the time when we talk about people's career path, they look at it as something that has happened in the past, and it seems straightforward to whoever is listening, but it's never as easy as you can hear it. So what were some of the hurdles that you had to go through in order to get to where you are today?
Well, there are several, of course. So first of all, there was another postdoc which I did in Melbourne with what is now the MIPS, but at that time it was not it was at the Clayton campus, at the Monash campus, the Department of Pharmacology, and I was with Roger Summers. And that was a very interesting period of time where I learned a lot practically, but we never published anything from that. It was a very short postdoc. It was a little bit less than 6 months. But career-wise, that was very important, and also grant-wise, because the grant agencies in Sweden had already always said, well, your work is, or your plans are good, but you have no experience from being abroad. And this short postdoc in Australia, together with my first bigger last author paper, which was published in PNAS the year after, in 2007, really made then the switch that I got the first 4-year position as a junior researcher in Sweden.
That's fantastic. Let me ask you, why Australia? How did that happen?
Oh, that's Roger Summers was a guest researcher, or he had I think a sabbatical in Stockholm, and that's where I met him. He gave a talk at our department, and we made some cytosensor experiments together here at Stockholm University, actually. And there the interest and the connection came up, and I decided to make a postdoc with him.
That's great. That's great. It's also very far from Stockholm to even Berlin to Brazil, but also Stockholm to Monash to Australia is quite a distance to go. And what were you focusing on at Monash? So my guess is molecular pharmacology, more signaling, but can you be a little bit give us a little bit context about your work?
It was actually a project designed to somehow compare signaling pathways of beta-adrenergic receptors and frizzles.
Okay.
So we had frizzled 2 there in focus, and with the lack of tools at that time, I didn't get very far on frizzled 2, I have to say. I managed to express it and to tag it and to see it, but we could not understand what we could do with it in the amount of time. So we did. I did quite a lot of microscopy there. I had a fantastic imaging facility on confocal microscopy, but signaling wise, we really didn't. We got stuck on the signature structure with frizzles.
Interesting.
Which we still do, unfortunately, sometimes.
Interesting. Yeah, because there's I think 10 frizzled receptors, and what is known about their signaling? And I know they're important in the word escapes me. Development. Yes, early development. Yes. Thank you. But what do we really know? If you met someone who knows a little bit about GPCRs, not much about frizzled receptors, and you had 30 seconds or maybe a minute to explain to them what we know, what we don't know, what would that be?
Well, it's a complex system with 10 receptors and at least 19 rather big protein ligands. And there's a bunch of co-receptors which are supposed to help the ligands to specify signaling. The frizzles look like GPCRs, but the field is somehow split in people who believe they are and other people who don't believe they are. So I think the biggest question is how we don't understand how the ligand interacts with the receptor, how the receptor really gets activated, and how signaling is initiated and specified.
Wow.
Unfortunately. And there are hardly any, or basically no small molecules available.
Interesting. So that's a lot of question marks there.
Indeed.
And so what do we know about frizzled receptors? What type of signaling they're involved in general?
Well, there are basically 2 concepts. First of all is what is sometimes called the canonical pathway, the Wnt beta-catenin pathway. That is defined by a Wnt binding a frizzled and a co-receptor called LRP5 or S6. And they regulate the stability of a transcriptional cofactor that is called beta-catenin. And that feeds directly into the transcriptional regulation of Wnt target genes. And for example, it regulates proliferation of stem cells or tumor cells. For example, very important in the renewal of the intestinal epithelium.
Interesting. So you mentioned tumor cells. So as many GPCRs or cell surface receptors, so frizzled are also implicated in cancer. What do they do in cancer? What's the issue with the signaling? Where does the signaling go awry?
Well, so the easy answer is that proliferation is regulated by an over exaggerated push towards Wnt beta-catenin signaling.
Mhm.
That can, for example, be due to mutations downstream of the receptors in a protein called APC, which is mutated in 85% of all colon cancers, and really pushing through beta-catenin stabilization the proliferation of the intestinal epithelium.
Mhm.
But you can also have upregulation of the ligands or upregulation of the receptors, the frizzles, for example, which will feed into an over signal. Or you can have mutations in any parts.
Interesting. Any particular frizzled receptors that you could call your favorite? I always ask this from everyone.
Yes, there are 10. Probably number 6, frizzled 6 is the one that I understand best because we have early on started working with it.
Mhm.
That I would probably say it's my favorite, but my other favorite is frizzled 10, and that is because it's so weirdly regulated. It's basically not expressed in the adult except for in regenerating vessels. So we don't really know and understand how it works. We have shown it's actually signaling through G13, which is still not understood what it's doing through this protein, but it's also regulating this beta-catenin pathway. And I think the interesting part, which we try to convince the community about, is that the Wnt beta-catenin pathway is G protein independent, but there are also G protein dependent pathways that are important, activated through Wnt and frizzles that might fine-tune the signal and the outcome, or even mediate motility, for example, through the 12-13 proteins.
Interesting. And then the other question I had, so you mentioned there's 19 Wnt and 10 frizzled. Any interactions or cross interactions between multiple Wnt and with a particular frizzled receptor? I wouldn't call it a promiscuity. I think it's not in systems like the chemokine receptor system. It's never promiscuous. It's very specifically and very well regulated.
So there are a bunch of co-receptors which might be important. I mean, it's an interesting question, especially today, because there will be a paper coming out next week where we use all human frizzles and map their binding to Wnt3A, which is one of the most famous Wnt's. And we show there are some frizzles that bind, some don't, and some are better than others, basically. So there's an affinity profile if you only look at the frizzles.
Interesting.
But for the 19 Wnt's, I cannot say that we know yet. It's always, in my presentation, it's always a big question mark.
That's so interesting. And do you think the lack of information is due to the lack of having the right tools to answer these questions?
Yes, the Wnt's are very particular because they have a lipid modification. And they're relatively difficult to purify and expensive to buy. I think half of my budget goes to buying recombinant Wnt's, and not all of them are available, so we cannot map, and they're differently stable, and it's a bit painful to work with them, and it's very difficult to compare those. But we are making progress, and now the binding story that we're working on was only possible because a colleague in Germany, Carrie Davidson, he managed to make a GFP tagged Wnt that has biological activity, and that we can use for a nanobed based binding assay.
Oh, that's fantastic!
But that's 1 out of 19. So we would like to have all 19 EGFP tagged Wnt's, of course.
Yes. In an ideal world, you wanna tag all the Wnt's and then be able to have different signaling pathways that you can very nicely measure. Obviously in a dose-dependent and a time-dependent manner, I think, and that the way I see it, it's like that beautiful map where you can just interrogate which Wnt with which frizzled and which signaling pathways get activated.
Exactly, and then a bunch of co-receptors in there as well. Yes,
of course. How many co-receptors are there that interact with
these LRP56 are the two most important one for the Wnt beta-catenin pathway, and then there are others, ROR 1 and 2, there's RYK and PTK7, and there are glypicans, which are most likely very important as well. So yeah, that's the main ones. But then there are also very interesting ones like some adhesion GPCRs, GPR124 and some others in the same cluster, which are very interesting as well. Functionally and mechanistically.
And then how come frizzled receptors actually have co-receptors? What does that? I guess it adds to the diversity of signaling and their ability to potentially bind different Wnt, different ligands. But how was that discovered historically?
Well, I think most of the knowledge from in the Wnt pathway was through epigenetic mapping. So the either in Drosophila or in frogs, they have been genetically mapped very early on, and these genes have been found to be essential for signal transduction. I'm actually not so sure that the definition of a co-receptor is well, I mean, it depends on how to define it. Is it co-binding the ligand, or is it actually really like would you imagine a dimer? So there, I think it is not necessarily that the receptors interact with each other.
Interesting. And so we don't know for these co-receptors whether they're binding the ligand. So do we know anything about the structure of a complex of a ligand with the frizzled receptor and a co-receptor?
Well, there are a few structures too, I think, of the extracellular cysteine-rich domain bound to a Wnt. But I don't think that's the full picture of ligand binding either. In this binding story that we're just about to publish, there we show, for example, that some frizzles, that the core of the receptor is also contributing to binding. So that is an interesting concept. That is not only this extracellular cysteine-rich domain. Mm-hmm. There's no full-length frizzled structure with the Wnt, unfortunately. We are waiting, of course, in the field. Will be very exciting. There will be soon coming a full-length frizzled G protein structure.
And so let's take a step back to the signaling. You mentioned that they couple to G13. Well, there's the beta-catenin pathway that controls expression of specific genes. But the G protein pathway, you mentioned G13. What other G proteins are known to interact with frizzled receptors?
The list is not complete yet, but we have started with frizzled 6, which we know is GI-GQ. Frizzled 5 is for sure doing GQ. Frizzled 4 is an interesting one because that's a very strong signaler through the Wnt beta-catenin pathway, which is G protein independent, but is then also coupling to 12 and 13. Frizzled 7 is doing GS, frizzled 10 is G13. The rest is basically not really mapped yet.
Correct. So it's all over the place, and it seems to be really dependent on which receptor you're looking at, the G coupling specificity. Exactly,
and I think that is one of the interesting advances now, or progress in the field, that we understand that a frizzled in a signaling system is not necessarily only the abbreviation FZD, it's also a number, because the individuals of the family are different. Mm-hmm. And I think that is not really particularly appreciated in the field because we didn't know so far.
And so you mentioned that there's 10 frizzled receptors. What's the homology percentage in between them, since they seem to be seen differently?
So the whole family is subdivided into different homology clusters, where, for example, 3 and 6 is in one cluster, and that is the one that is closest to Smoothened, which is also a nice, interesting player in the whole class. And then there you have 1, 2, and 7, and 4 and 9 and 10, and I think 5 and 8 different homology clusters, where the homology is higher in one single cluster. And they seem to behave more likely among a cluster than across the clusters.
Interesting. They seem to be really. There's enough work for everyone to go around and try and better understand these GPCRs. One of my questions is, what do you think are tools or initiatives that are lacking? What do we need to better? I'm sorry, it's Saturday. It's been a long day. So my question is, what are the tools that we need to develop or that are lacking currently in order to better understand the function and the signaling of frizzled receptors?
I think there are 3 things that are very important. First of all, I think the field needs to understand that these receptors contribute not only the Wnt beta-catenin signaling, but also G protein signaling. Mm-hmm. And if a more open mind would develop, that would help to grasp the whole picture. That I think it's conceptually one thing that is important. Then tool-wise, I think pharmacological tools would be very, very attractive. That we have small molecule ligands that act as agonists, antagonists, inverse agonists, preferentially with an isoform or subtype selectivity, or at least with a cluster, homology cluster selectivity. That would be fantastic. And I think it's in reach. We have recently shown that small molecules can be used as ligands in frizzles. That was unclear, especially from the first frizzle 4 structure was proposed that a ligand binding pocket was too small, but they actually can be targeted, and that will be an interesting development in the next coming years. And then, of course, structural information of a full-length frizzles. In complex with the ligand, in complex with the co-receptor, in complex with the G protein, and so on, in complex with disheveled, so on.
And perhaps more GFP tagged Wnt, and the technology to actually make them and be able to purify and have all 19 of them ready
so the problem is not necessarily the making of it. The problem is to be lucky enough that they actually be biologically active.
Yes. Well. That is true. That is true. You absolutely need them. How big are Wnt? How big? They're lipoproteins.
Exactly. They are about 35, 40 kilodalton. So they're big.
Pretty huge in that sense.
And they have these lipid modifications which somehow pose an interesting conundrum, because they cannot really diffuse through the aqueous medium. They need a carrier protein, they need a membrane, or they need a lipoprotein particle to be transported. And then a thermodynamical conundrum for me always is how a carrier protein really hands over a lipid moiety to the receptor to bind the extracellular cysteine-rich domain. That is, but biologically can make sense out of it. We don't really get it yet completely.
Any carrier proteins that are known already in the humans or in animal models that actually transport the ligands?
Yes, it's mostly like serum proteins. So if you have cells that secrete Wnt, you will not get them into the medium without serum. So serum proteins work, and there's a specific one. It's called affamine, which has been described as also in a structural way as a Wnt binding protein.
Interesting. That sounds like a really difficult class of receptors to work on, but at the same time very fascinating. Where there's discoveries to be made, but there's also a need to develop better tools, like tool compounds that you mentioned, to better understand. What type of tool compounds? You were mentioning small molecules, and that it was shown that there are small molecules that actually bind frizzled receptors. What's known about these tool compounds or potentially small molecules?
Yeah, so I mentioned Smoothened before. There, the pharmacology is a bit richer. The cyclopamine is binding as an inverse agonist to Smoothened, which and there are small molecule agonists which are called SAG, Smoothened agonists, and those ones we could, at least one SAG 1.3 we could repurpose for frizzle. We could show that it also acts at frizzle 6 as a partial agonist, a very weak partial agonist, but it does it. So that's of course a starting point to think how one could target these receptors.
Any chemical modifications worth doing on this particular compound in order to, transform it into an agonist, a full agonist, or at least a stronger partial agonist in the plans?
If I'd only knew. So far, we are very successful to transform into an antagonist, but
okay,
but not not so far.
That's still progress. At least when you have a molecule that binds and that does something, that's already a good start in that sense. So, I'd like to take a step and ask you a couple of questions about the disease areas. We've talked about the importance of frizzled in development. But also in cancer, any other disease areas that, mutations in the Wnt or anything that is in relationship with the frizzled receptors would cause any other diseases?
Yeah, there are, I think, well, I could mention fibrosis, for example, which is lung fibrosis and liver fibrosis and also heart fibrosis that have been linked to Wnt signaling, not necessarily a specific frizzled.
Okay.
There's one interesting finding is that, for example, Frizzled-4 is very highly expressed in fibroblasts, which could of course be some kind of a link, but has not been explored to my knowledge. Interestingly, mutations in frizzles are very rarely linked to. Our start with Frizzled-6 was based on a Swedish geneticist who found that Frizzled-6 mutations were intrinsically linked to nail dysplasia. And from those mutations, we actually started working on Frizzled-6, where we for the first time could see that there's I hardly dare to use the word a bias, a functional selectivity in a mutation towards disheveled and G proteins in Frizzled-6. So that can be mutationally regulated, and these mutations are having a human phenotype. It's not particularly dramatic. You have nail problems. The frizzled knockout mice are also losing their claws after a while, so it's reminiscent to that phenotype. And the interesting part in Frizzled-6 knockout cells shown by Jeremy Nason is that these mice are whorly, so they have a very interesting circular arrangement of the hairs, the fur, basically.
Interesting. Wow.
So it regulates planar cephallarity in two-dimensional tissues.
Wow, who would have thought that knocking out Frizzled-6 would have an effect in the hair? You mentioned that Frizzled-10 is only expressed is not expressed in the adult human.
Right. So it's interesting if you look at cells, like it's established cell lines, Hex cells, Show cells, they often have many frizzles, but they hardly ever have Frizzled-10. And it turns out that it's expressed in the brain vasculature during development, on peaking, but it's also regulating the placenta, for example, in the regenerating vessels there. So it for sure has something to do with vascularization and regeneration of that.
Do you think that maybe it stops being expressed in the adult because of potential activity, signaling activity that's constant, that's selective, and it might have a bad effect if it were expressed in adults, and it would potentially lead to cancer? I'm just wondering, because you mentioned
it's possible. Yeah, I mean, we are starting to understand, or at least to really starting to understand constitutive activity of these receptors as well. So we've seen it for Frizzled-6 very weakly before, but we have others for example, Frizzled-7, which seems to be highly constitutively active towards GS, and it works for like Frizzled-7 G protein interaction, but we have no idea what that component of constitutive activity towards GS means in a physiological context.
Yes.
Since we don't have any inverse agonists, basically.
Yeah, and so we talked very, very little about functional selectivity and bias, and then the natural question is where do beta-arrestins come into the story? Is there any effect of beta-arrestin?
Yeah, so this PNS paper that I was mentioning very early, my last author paper, the bigger one, that was about beta-arrestin in Wnt signaling. So it is interesting and weird that beta-arrestin is really necessary for Wnt signaling. It is not really clear if and how it interacts with frizzles. It for sure interacts very strongly with a downstream element in Wnt signaling called disheveled. And that was, among others, shown in this paper and has also been pointed out before. And it's not only necessary for Wnt beta-catenin signaling, but also for beta-catenin independent signaling to diverse pathways.
That was one of my next questions, as to where we've talked about G protein dependent and independent signaling of frizzled, but then where would beta-arrestin fit in there? And I have very vague memories of beta-arrestin 1 being important in the beta-catenin signaling, and I may not remember this correctly, but I think there was at some point this divergence between beta-arrestin 2 function and beta-arrestin 1. Oftentimes, when you measure signaling of GPCRs and you try to compare beta-arrestin 1 versus beta-arrestin 2, I'm thinking about the beta-adrenergic receptor and chemokine receptors I worked with, you would get for the same ligand, you would get potentially the same effect, but at different. I'm really looking for my word here, at different levels. So beta-arrestin 2 signaling was always much higher than beta-arrestin 1. And it's very difficult to separate the functions of beta-arrestin 1 versus beta-arrestin 2. Any indication that one of the two arrestins has a better effect or a better signal transducer on frizzled receptors or Wnt signaling?
Not that I really know. So in the earlier studies, which I referred to before, we had the knockout maps, and there was no really difference in beta-arrestin 1 or beta-arrestin 2 knockout maps. They were both inhibited, and in the full knockout, it was stronger. The phenotype was stronger. Mm-hmm. So I can't say. But at that time, we only looked at disheveled and beta-arrestin interaction.
Got it.
With regard to frizzles, we have not really looked, I have to say.
I wonder if there is, kind of beta-arrestins being kind of like not the mediator, but the molecule that is preferred to interact with the receptors, or at least mediate receptor signaling in the absence of or presence of G proteins, depending on any preferences.
In the case of frizzles, it's a bit more complicated because there's a third one, which is called disheveled. And that one is in the way. And I think it might also be in the way for G proteins. And I think one of the biggest questions to solve is how the receptor might define signaling through disheveled over G proteins. And this might be regulated through subcellular concentration of one or the other. But we think now that with recent data, in part unpublished data, that there's a conformational selection of also disheveled towards frizzles. So that is, I think, very similar to a conformational selection of G proteins.
And would binding of Wnt or binding of ligands influence that conformational selection?
I think it does. It is a bit tricky to measure in a way that when you overexpress disheveled, it is already stuck at the receptor. So okay. It is very tricky to see differences with Wnt addition. With that being said, manuscript submitted, so we will see how the community takes that piece of information, hopefully soon.
Any differences in localization or tissue specificity of expression of Wnt versus frizzled receptors? Would that also give an indication onto their differences between the function of the receptors in different tissues?
Absolutely, that's very important, and that point is often forgotten when you stimulate frizzles in HEK cells, because there's in the developing embryo is of course a very complex arrangement. You have polarization of cells, you have asymmetric localization of the receptors, you have even selected asymmetry of co-receptors versus frizzles, for example, and you have also a gradient of the ligand. So and those minute regulatory mechanisms, they of course get lost in a HEK cell, and from that point of view, it's very important to also integrate the knowledge from like a purely pharmacological experiment of a ligand binding affinity into the physiological context of a proliferating epithelium or a developing embryo.
We talked about HEK cells, and that this is one of the questions that comes out. My guess is you're using HEK cells, and you mentioned that you have different frizzled receptors expressed in HEK cells. What is a good cellular background, a lab type cell that you could be working in, where hopefully you can just, express in one receptor or two receptors together? Or is there a system like that?
Yeah, well, we have been looking desperately for cells which only have one frizzled. Any physiologically relevant cell that we have been looking at, for example, the mouse primary microglia, they have like 5 or 6 different isoforms on the RNA level. Antibodies most likely would not work nicely. Yeah. We have been working with Jeffrey Rubin at the NIH, who found a homeopathic, you know what I mean, cell that is called 32D cells that had no frizzles, and he would stably retransfect. And that was a breakthrough. But they were very difficult to handle. You could not really transfect them transiently. And a friend of mine, Vijay Bria, in the Czech Republic, we did postdoc together. He said when CRISPR came up, he said, "Oh, you have to do the full class F knockout CRISPR hack." I said, I would like to have it, but I'm sure someone else will be faster.
It's complicated, yeah.
Yeah, and someone did it, and that was Benoit van Hollebeek in Belgium, and he was generous to share this cell line with us, and it's a blast. We have so much fun with that cell line to finally be able to work on a frizzled null background, and to be able to retransfect an individual frizzled and then study that with regard to function and binding and conformation changes, and that has really made a difference. We don't always use it, depending on the assays, of course. If we have a fluorescent frizzled, we only look at that. It might not be so important, but it has been a fantastic tool in the recent. I think it came out 2 or 3 years ago.
That's great. That's why I was asking, because I also, HEK cells are great. You can culture them; they grow like grass, and very quickly you can have a lot of cells. But then I also encountered an experience when I was trying to understand S1P receptors, and it turned out that out of the 5 S1P receptors, there was 3 or 4 expressed in HEK cells. So it was kind of difficult to measure anything that wasn't labeled, because if you're doing Fred or bread or nanobread experiments, and the 2 molecules that you want to look at are labeled, then it's easy to quantify that interaction. But whenever you're trying to look at a signaling event downstream, I'm thinking GS or GI activation in cells that do have endogenously the receptor, it's kind of hard to differentiate those.
Indeed. I mean, the problem is now we're getting greedy and would also like to have the G protein knockout on the frizzled null background and the beta-arrestin knockout and so on. So, but
yeah, it's a lot of work, but I think at the end, as you mentioned, it's a really great tool, and I think it might be worth investing and collaborating with labs who do this, with their eyes closed. It's not difficult; it's just time demanding.
Exactly. Yeah.
To do this, but
we have actually managed to also knock out Smoothened from this cell line, so we have a class F knockout. So that's as much as we could do.
That's great. And how does it change anything about the cells themselves?
Yeah, they don't like life equally much as their cousins. Yeah.
Okay.
They grow slower and they're a bit tricky to thaw, and yeah. So they are not the same as before, which is not surprising.
Of course, which is a good thing. Do HEK cells by any chance secrete any Wnt or any ligands?
Yes.
There you go.
They secrete at least Wnt5a, but probably several, which has been a problem. And now, especially if you work on constitutive activity, it's a problem. But then there's this beautiful compound, or the beautiful compounds. They're porcupine inhibitors. Which porcupine is important for the secretion of Wnt, and if you inhibit that molecule with a very effective chemical and very potent chemical, small molecule chemical, then you can blunt the secretion of Wnt, and you see that the basal signaling goes down, like for example on a protein mobility shifts. So you can be relatively sure that you have no endogenous ligands around.
Wow.
So that in combination with the frizzled null cell line is really a treat for us. Yes.
That's fantastic. And what kind of methodologies do you typically use to interrogate frizzled and Wnt-frizzled receptor signaling with these cells?
So originally we've done a lot of Western blot with phosphor-specific antibodies for the LRPs, for example, and beta-catenin. We have also generated a phosphor-specific frizzled antibody, frizzled-6, at some point, which was exciting because it showed that the phosphorylated form of the receptor is asymmetrically distributed in fallopian tube epithelium, whereas the unphosphorylated form, or the total receptor, is equally distributed. But we've initially, when my lab started, we did a lot of confocal microscopy and live cell imaging. We adapted, for example, this very time-consuming double-color FRAP method from Nevin Lambert to show that there's a complex assembly of G protein and frizzles, which will be dissociating when you add Wnt. But that every single measurement or point on a graph is a few days of work. So the recent years, we have adopted a lot of bread assays and developing own bread sensors or fluorescent sensors for that part. So we do plate reader assays on mostly bread. which increases our throughput and also makes things like dose response curves actually feasible.
Yes, and time course as well, which kind of can be
exactly.
You could do both in the same plate, and then the difficulty becomes analyzing all the data points. But that's easier than having to stay and look at the cells under the microscope.
Indeed, yes. Yeah, that technology also with the people in the lab has really evolved our thinking and our way of designing assays and looking at protein conformation as a readout of changing in fluorescence, like with these CP-GFP conformation sensitive GFPs, where we clearly see that Wnt stimulation changes the conformation of the frizzles that we have looked at in the same way or similar way as the GPCR acts. So that is my strongest argument for the GPCR nature of these receptors.
And then I think it's also a nice way to prove it, or to kind of make a point to the GPCR community and say, well, although the frizzled receptors can interact via G protein to end, the beta-catenin pathway, the fact that they move and they change their conformation when they bind their ligand similarly to a GPCR is an additional clue that they are GPCRs most likely.
Exactly, and I don't see why nature would invent a ligand receptor system that works that way, but would not utilize it.
I think nature is really smart and really tricky, and I love the idea of, having a system that works already and just fine tuning it in order to get a broader sense of signaling. I think being able to couple a G protein is very important for a GPCR, but also being able to signal without the G protein. Now we know that is very important for quote unquote regular GPCRs, but also for frizzled receptors can be very important as well.
Yeah, I mean, given the numbers of receptors of GPCRs, sometimes I wonder how regular a beta-2 is in the way that it really is so robust. Other orphan receptors are not. So, and frizzles are not as well. So, I mean, we have a hard time to work with these receptors. It's not like in the good old adenosine receptor times that we have the small molecule available and get crystal clear tenfold increase of a MAP kinase phosphorylation or something.
Mm-hmm. Yeah, they're similar. Yes, of course, at room temperature. I think there's a lot of questions that remain to be answered, not only in the realm of the frizzled receptors, but also for a lot of orphan GPCRs or orphan receptors that look like GPCRs, where we don't know what the ligand is, but you can fantasize and try and come up with an explanation as to what they're doing and how they're interacting with the intracellular signaling pathways and what type of ligands they're binding. And I think nature is really smart. So if it's there, it must be doing something important.
Feels reasonable in the right context, of course. Yes.
Yeah, of course.
And I mean, I think constitutive activity is also very important, or might also be very important. And then its negative regulation could be physiologically relevant, for example.
That too. Although I don't know how many examples of constitutively active receptors there are in nature. I think it sounds a little bit more dangerous than it is, depending on the function of that receptor. But I don't know. To me, it sounds dangerous to have some receptor in your body that's constitutively active, unless it is very important. But still, it could have definitely some physiological relevance. So,
Indeed.
what are the next big steps, next big questions that you plan on asking in the next, let's say, 1 to 5 years that you really want to get an answer to?
Yeah, so it is in part is the physiological role of the frizzled G protein axis. There are, I mean, we have my lab started originally working on microglia cells and the role of Wnt in there, and we could easily measure GTP gamma S binding induced by Wnt. So. And that was important for the pro-inflammatory regulation of microglia. And that is, of course, important for glioma and for many other things, which are in turn regulated by high levels of Wnt proteins. So I'm sure there is a physiological relevance. It has been shown in muscle development for frizzled 7 and GS and for other combinations as well. But that is of course one thing that is burning. But since my lab is more biochemically oriented, I think for me and for my lab, it will be understanding the conformational changes that lead to pathway selectivity. So which Wnt will do what in order to signal through a G protein or through disheveled. I think that selection of pathway selectivity is very exciting. And then, of course, in the next round is the question if we can target that with small molecules in a classical functional selectivity approach.
Mm-hmm. Yeah. So basically, you want the map, and you want to be able to follow which ligand with which receptor does what, and hopefully link that to a pathophysiological or physiological context.
Exactly. I mean, the recent development in my lab has gone a bit towards structure and in silico work as well. So mm-hmm. we do a lot of molecular dynamic simulation to really understand the dynamics in the system. Yep. And I think that the mechanisms that are behind the activation of Wnt beta-catenin signaling towards the G protein and also other disheveled dependent pathways are, of course, intrinsically different. And as I said before, the conformational selection might be a key in that.
Agreed. I can very, very, very much see it being the case. Having one receptor, one protein on its own, or a complex of proteins that and their ability to bind the third protein definitely would be changed depending on what's that conformation that they're adopting, and what's that state that makes sense for them to be in, and that definitely, I think, that determines the signaling pathways that might be activated.
A few years ago, I said when I understood the frizzled G protein coupling, I will do the frizzled arrestin coupling.
Blink.
But we're not there yet. So,
yeah, perhaps when you get to the point where you have the knocked out hex cells, the F class knockout hex cells, and then start removing different G proteins, and also start removing one or both arrestins at that point.
Yeah, maybe.
I don't know if the cells would how would the cells feel about that, removing all those proteins, all those.
Yeah, it's molecules. Interesting, Sam. I mean, at some point we thought that cells without disheveled wouldn't survive, and they survive as well. So it's difficult to predict. The cells they rewire. I think that's the term to use. Exactly.
I think I was having this discussion about knocking out a specific G protein in hex cells, but then the cells rewired and found another way of another G protein and another way of surviving. So at that point, yeah. But at least it's a good tool to have in the lab, and being able to selectively ask these questions is really crucial to better understand receptor function.
And I mean, this technology is also crucial to transfer then the assays maybe to cells that endogenously express these receptors, or to organoid studies and to approach the physiological relevance in simplistic models. That's another attractive line.
Yeah, I think hex cells in general are very valuable, just as valuable as a good tool compound, because they allow actually the interrogating and asking these basic questions, and then taking that information and putting it in a larger context, as you mentioned, is more physiologically relevant cells, but also more physiologically relevant systems like a knockout mice, for example.
Exactly.
And even higher. I asked this from everyone, and considering that you've traveled around, you're originally from Germany, and you spent some time at Monash and everything, what would be your advice for junior scientists who would like to contribute to the field?
Yeah. So, and junior, you mean?
PhD postdoc.
I'm thinking about my oldest son. He's turning 18, and he's about to choose what to do after high school, and. Mm-hmm. I think the scientific motivation in him is, of course, through the excitement at the dinner table. Just clearly how we handle discussions about whatever biologically or physically or something like that. And I think that is crucial also to keep the excitement going. And that is especially important for young scientists. And despite all the obstacles with grants, with fundings, with difficulties of the experiments, if you keep the fun of it, I think that's my most important important advice. I think I've maintained it despite rejections and things like that. But keeping the fun and the curiosity in this in biology. And I think if it doesn't really matter if you, as I said before, if you end up in virology or in pharmacology, as soon as you scrape the surface, you really and you find the unknown, then the excitement comes up. If you have the right personality. But practically, I think what I have seen works for myself and also for others is that if you have a PhD project and you, for example, run a postdoc, and then think of in which direction to take off towards an independent career.
Yeah.
I think if you make a reasonable merger and find your niche very early, that is important. And then you need, of course, to have some kind of a stubbornness to possibly fight windmills that will always appear. I had an interesting discussion with Martin Lohse when I was getting independent after my second postdoc. My projects were all over the place, basically. I was doing neuronal retrograde transport of arrestin receptor complexes, NK1 receptor, a little bit frizzles, a little bit wins.
Mm-hmm.
And I was about to, I got money in for that, but relatively small grants, but money was coming in. But we had at Karolinska from the junior faculty a grant that is called Visit the World grant and invitation grants. The invitation grant was that, and in that way, Lohse came to Stockholm. He was invited to give a talk and then to evaluate my research. And we were two who applied in that position. And he said, "Well, you can't do this. You cannot be so unfocused. You need to make up your mind." And then I focused on frizzles, and that was a very wise decision. But that also really merged my PhD experience and the postdoc experience to something that is relatively unique from the approach. So I think that's a good one for young scientists.
Yeah, I think focusing is important, but then again, I think before getting to focusing, exploring like you did, working on adenosine receptors and going to Australia and learning new things is also very important because it allowed you to broaden your horizons, but at the same time figure out what was really what you were passionate about.
Exactly. Yeah.
And I think that's very important to do both. Go wide, and then once you feel like you've seen enough, then focus on what really gets you out of bed and what you feel like it's fun to do every day.
Yeah, and then of course, if you manage to ask the right questions, the problem in my field was often that the tools were not available to answer those questions, and I've been jumping in between techniques that were relatively difficult to set up, like these microscopy-based double color fraps and FCS and stuff like that. And then I've answered questions, and I went along to the next technique, like setting up the bread stuff and developing new biosensors. So for me, it's important to not work on a technique, but rather apply techniques to answer questions. That can be very painful in setting up, but then also letting go, for example. I mean, it's interesting how the laboratory character changes with the people that come in and know a technique or know to push something, and then after a while they move on as well, and then there would be another different flavor somehow.
You make a great point about not focusing on the technique, but focusing on on the questions and what techniques will help you answer those questions. Because there's a lot of things out there, and you mentioned the difficulty of setting up a new technique. I'm sure it's not easy, but if you stick to it, and it's like adding a chord to your guitar, and then when people move on, the technique and the technology stays in the lab, and then you have a new cohort of students coming in and postdocs, and at that point they get to learn something, but at the same time they could be bringing new technologies that would help answer some of the questions you're interested in working on.
Indeed, yeah.
What were the top 3 aha moments as a scientist that you had? And I'm interested in it can be science-based or any interactions, like you mentioned your chat with Martin Lohse, who said you need to focus. Anything that changed your way of thinking about science?
Well, I think the one that I'm immediately thinking about is just very early in undergraduate studies, when we for the first time purified or isolated bacterial DNA. That was so cool. It was just a slimy clump around a wooden stick, but that somehow ignited a fire. And then similar to that, the next one was the when I came to Stockholm and perfused my first rat and isolated the whole CNS brain, including spinal cord, dissected it out. That was, as a biochemist, for the first time really doing something in vivo, even though the rat was dead. But that was exciting and also triggered something in me. And I think that was for me as a biochemist from Berlin coming to Stockholm was really important to have this at the Karolinska Institute, which is a purely medical university and with a very close connection to the clinics. That is still something that I value very much, even though we don't do the clinical collaborations all the time, but the translational aspect is always there. And then something that has helped me over the years a lot is a third anecdote, which is from my PhD. I mentioned before that you do a half-time seminar, and I had 2 manuscripts ready that were about to be sent after this seminar. And I was using pharmacological inhibitors to dissect the signaling pathway. And I came up with something weird, a relatively unknown connection of something to GS to PI3 kinase or something like that. And in this seminar, my supervisor, Bertie Friedam, asked me the questions, have you checked if the inhibitors are antagonists at the adenosine receptors? I said, no, why would you do that? It's obviously that an ATP site inhibitor has some kind of structural resemblance to an adenosine site of a receptor. And it turned out, and this was then, I skipped both manuscripts, but it became another manuscript in my thesis, that several of these antagonists or kinase inhibitors were actually antagonists at the adenosine receptors. And that's why I saw an effect. And that provided me a relative humbleness towards chemical probes and inhibitors. And I think I've learned my lesson there. The hard way at the time, but it left traces.
That's fantastic. That is really fantastic. And then my last question to you, and I ask this from everyone, if you have availabilities for positions to work in your lab, how can people find you?
Yeah, I think it's relatively easy to Google my name and connect that with Karolinska, then it will pop up. But at KI, at Karolinska Institute, we are obliged to, if we have a position, like a PhD position, for example, we are obliged to announce that on KI Jobs. So that's the central homepage of the institute, of the university. And I would, for PhD positions, always do that. Otherwise, I'm announcing my positions on Twitter, at Schulte Lab, on Twitter or LinkedIn, for example. That's what I usually do. Otherwise, if there's interest for whatever rotation students or postdocs, just drop me an email. That's the easiest way. Exactly.
That's perfect. That's perfect. So the links to your LinkedIn profile, your Twitter, as well as to the Karolinska KI's website will be on our website when we release this episode, so people can go there and easily find you.
Perfect. Great.
Thank you so much, Gunnar. It was great. I really appreciate talking to you. I learned a lot. I hope the audience also learned a lot about F frizzled receptors, and I think there's a lot of work to be done. It's exciting, and hopefully at some point we'll have enough tools to answer all those questions and have that signaling map up and running and ready to go to share with the world. I'm pretty sure it's going to be a bunch of manuscripts, but still, I can see it. Having that map on a wall and being able to follow those events, at least the majority of those events.
Sounds promising. Thank you very much.
Thank you so much. Have a good evening. Thanks. Bye.